Bacteria. We'll come back to bacteria at the end of this section. We really know nothing about transcription replication conflict in archaea at the moment. Hopefully we'll publish something in that soon and I can talk about it next year. But I want to talk a little bit about eukaryotes. And this is just two extremes within the eukaryotic tree of life. This is what I think a human looks like. This is a trypanosome, and I'll tell you a little bit about the two different organisms. And you've seen some of this data already. If you'll remember, this is marker frequency analysis. Remember the gene dosage, identifying replication origins within trypanosomes. So trips are bizarre organisms. They have these really peculiar chromosomes. Their transcriptional regulation is highly atypical as well, because what they have are large blocks of tens, if not hundreds, of genes that are transcribed as a single polycistronic unit. A single transcription start site, and then many genes are transcribed as a single transcript that is then processed by an event called trans-splicing, where they chop it up and add a little cap to all the messages. But what this means is that large chunks of the chromosome are transcribed in one orientation. So the grey blocks indicate genes, and above the line is left to right, below the line is right to left transcription. So we can see, for example, an origin of replication here transcribes this direction. Another origin replication down here, transcription going this direction. And the point to notice is we see the peaks for replication origins. There's a slope on both sides of the peak, so this is classical bidirectional replication. But hopefully what you can see is that the slopes have different gradients. We saw this with the bacterial system before. Remember Jade Wang's data with Bacillus when she makes transcription go against, or replication go against transcription. You see a change in the angle. That's what we're seeing here. Here's an origin of replication. The fork that goes in this direction is initially going with the direction of transcription, surfing the wave of transcription, if you like. The fork that's going in this direction, however, is battling transcription that's coming in against it. So in a given period of time, this fork goes further, the slope is shallower. Then this fork here is steeper, is conflicting with transcription. Okay. So we can see in a genome-wide level that the very organization of the trip genome with these bizarre big polycistronic units really is biasing things towards different parts of the chromosomes being replicated at different rates. Now, is there a regulatory component to this? We don't know. Maybe you could imagine you might want slower replication, so you'd set it up in this orientation. I don't know, but the fact is clear that you can see different replication rates purely because of this conflict with transcription that's ongoing. Okay. Just jumping back, this earlier slide, we can see in many cases though, for example down here, this late firing origin, transcription is largely collinear with replication. Okay, so the origin, it goes this way on the upper strand and then this way in the lower strand. And that's concordant with the directions of replication. Similarly here, we see an early origin initially starting out the same direction as transcription. Same in that opposite strand. Okay, so yeah.
What is this method called, or how did they
Marker
get this?
frequency analysis. So in trips we do have large chunks of the genome that are replicated in conflict with transcription, but generally eukaryotes seem to prefer to avoid transcription replication conflict. And there's one specific example that's been studied in quite nice detail in a number of different organisms. And that's in the ribosomal RNA locus. So, as you all know, this is transcribed by POL1, RNA polymerase 1. And ribosomal genes are very heavily transcribed. They're generally found in arrays. Those arrays are present in a special nuclear substructure called the nucleolus. And we see this repeated structure of the array with the 35S ribosomal RNA precursor, the 5S ribosomal RNA, and another 35S ribosomal RNA. The 5S and the 35S are transcribed in divergent orientations, and in many species, including budding yeast, fission yeast, and mice, and presumably us as well. Replication origins are found in the area between the 5S and the 35, or in case of humans, 40, or mouse 45S ribosomal RNA. And that's nice. You've got replication starts here. It goes collinear with transcription, but it's in a large array. So if replication continues out of this region, it's going to encounter heavy transcription coming in the opposite orientation, in the conflicting orientation from the next array in the next unit within the array. And so what eukaryotes have is a replication fork barrier. They've actually evolved a system or systems where they stop replication specifically at this site between the 5S and the 35S ribosome RNA prior to the conflict happening. So they deliberately set up a roadblock within their chromosome that prevents transgression of a replication fork into the converging ribosomal RNA locus. And that's just, in the case of budding yeast, a simple little sequence found in the chromosome that serves as a binding site for this protein here, FOB1. Rather intriguingly, in fission yeast, the same principle exists, that we have these fork barriers, but they're bound by distinct proteins, REB1 and SAP1. Furthermore, even in yeast, we see possibly a distant relative of SAP1, but largely a distinct protein binding to a single site within the mouse locus. Okay. So the principle of having fork barriers present to prevent transgression of the fork into the non-permissive zone, if you like, is conserved, although the actual proteins and cis-actin sequences are not.
So how does it get replicated?
Good question. Generally, you'll see an example in the E. coli system where we know the definitive answer for that, so I'll tell you that. Generally what happens is that the fork barrier is actually polar in nature. So it will stop a fork coming in this orientation, but it won't stop one coming in this orientation. As far as I'm aware, the basis of the polarity of the barrier is not resolved for the eukaryotic system. It's very clearly resolved for the bacterial systems, and that's what I'll tell you about in a slide or two's time. Yeah, otherwise you would have a problem, because you would have, you know, you'd come to this side, you'd replicate to here, you'd stop, you'd replicate into here, you'd stop, and you'd have a little bit that's unreplicated.
Yeah.
That's why I'm not using the laser pointer, I have no idea. But yeah, there would be a major problem. And just some experimental data showing this is our good old friend, the 2D gels. If you remember the replication fork arcs, the Y arcs, and if you have a species where the Y arc is, where the replication fork is paused at a specific site, then you'll see accumulation of that signal. And there we go, in the presence of FOB1, we see this in the interstitial repeats of the ribosomal locus. If we delete FOB1, now we've just got a smooth arc. That stalling is gone. So this protein is imposing the barrier at this position. Folks have done nasty experiments to yeast. Yeast gets abused really, really unpleasantly by a lot of folks. And what they've done is they've taken the FOB1 binding site and they've stuck it at ectopic positions within the chromosome to set up an ectopic barrier to the replication fork. And again, this is another important point to emphasize, that these things are not permanent, forever arrest sites. Ultimately, the replication fork is going to come up to it. It's going to encounter it, it's going to try and push through, it's going to fail. It'll try again, it'll try again, it'll try again, and eventually it's going to win. Thermodynamically, it will eventually manage to push through this barrier. Okay. We have accessory proteins that are brought in specifically to stalled forks. We saw that in the case of yeast, of E. coli. Remember the accessory helicases, the Bugatti Veyron and all that stuff, the rep protein, the DNAB, one in one strand, one in the other, turbocharging through the barriers. Well, eukaryotes use exactly the same principle. We have replication forks approach the barrier, they stick, the fork actually stays together. Okay? The fork doesn't dismantle. The cell's made an awful lot of committing steps, getting an active and functional replication fork. It'd be crazy if this thing just fell apart. And so there are checkpoint systems that requiring certain proteins, more specifically TOF1 and CSM3, that help this stay in a sort of resting, sort of stasis position almost at the fork. It doesn't just dismantle. If it is stuck, though, eventually what it's going to do is it's going to bring in an accessory helicase, a thing called RRM3. This is just like the situation that we saw with REP, where we're now bringing in a helicase functioning on the opposite strand that will help to drive through this barrier, and the very act of melting the DNA, this turbocharging by the RRM3, will help to displace the FOB1 barrier, whereupon the fork can recommence upon its way. So RRM3 eventually is brought in to stalled replication forks to essentially turbocharge their way through issues. So again, a concept shared between bacteria and eukaryotes. Even though the proteins are different, the fork assembly is different, even the polarity of the central replicative helicase, the MCF complex in this case, is different from the bacterial DNAB family. Bugattis are universal. Okay, jumping forward to higher eukaryotes. This unbelievable, to me it's just a mind-blowing observation that they're, yes, Raj, save me from the unbelievable.
Oh, sorry, one question.
Please.
So the FOB1, how strong it binds, like how length of the DNA it binds?
I have absolutely no idea. Great question. Don't know. I don't know its affinity. I don't know the size of the binding site. Somebody can Google it right now. It's probably out there in Wikipedia. But no, I'm sorry, I actually don't know. I failed you. There you go. I flunked my exam. Now I know what one to ask. What is the footprint of FOB1? Yeah, don't know. Higher eukaryotes, they have genes that in many cases can be as big as a prokaryotic chromosome. Just mind-blowing concept. And in terms of the actual amount of time it takes to transcribe a single gene, it can actually take longer than the cell cycle itself. Hours and hours and hours to transcribe a single transcription unit, like Duchenne muscular dystrophy is 2 megabases in size, the gene for that. And what was noticed that in many cases, these massive genes can correlate with sort of fragile sites prone to breakage in the chromosome. This is just a beautiful experiment that illustrates that. Here we've got some genes, and I don't even know what CNTNAP2 is. I have no idea. If anybody does, tell me. It's a gene that's transcribed in the grey, in myoblasts, but is absent in the black, beta lymphoid, okay? Conversely, this highly memorable gene here, IMMP2L, trips off the tongue beautifully, highly transcribed in beta lymphoid, reasonably highly transcribed in myoblast, whereas WOWOX is in beta lymphoid but not in myoblast. Okay? And this is now some sort of chromosome painting type experiments where they've taken fluorophores on one side or the fluorescent DNA that will hybridize to a locus immediately on one side or the other side of this individual target genes. Okay? And what's absolutely lovely is if you look at the FHIT, this one here, which is transcribed in beta lymphoid but not in myoblast cells, then we can see a clear physical breakage in this particular chromatid. Okay? Don't see that in the myoblast, it's contiguous sequence. Transcriptionally active, we get the break. Transcriptionally silent, we don't get the break. We look down here at WOX, then again in the beta lymphoid, we see breakage, in fact loss of the telomere from this chromosome, almost, whereas chromosome integrity in the myoblast. DMD, myoblast enriched, we see the break here, we don't see it when it's not transcribed in the lymphoid tissues. Okay? So transcription's correlating with the observable physical breakage of these sister chromatids. Something about transcription is leading to these being a problem. And the prevalent model, which, to be honest with you, I absolutely bloody hated for the longest time, it just seemed like a farcical model, is that what we're getting is, yes, there's a clash with transcription, but it's not really the machineries as such, but rather it's a byproduct of transcription. And that's the generation of what are known as R loops. And so R loops are where the messenger RNA itself invades the double-stranded DNA and sets up a rather stable RNA-DNA hybrid molecule. And that then serves as a really robust roadblock for a couple of reasons. And part of the reason I always thought this was a farcical idea, and I'll tell you now, I'm completely wrong. But I thought it was a farcical idea. It was because of cartoons like this. Okay. Because you guys now, you've had Craig's lectures. You're experts in transcription. What's wrong with this?
It's a much bigger machine.
The RNA polymerase is bigger, but think more specifically about how they're drawing the DNA here.
It's reversed direction of the helix. Yeah.
We can turn it around. More fundamental than that, in transcription,
The supercoil.
supercoiling, we'll come to that, yep, absolutely, but even more fundamental. How big is the melted region? Where is the melted region during transcription? 20 to 30 nucleotides. So it's entirely contained within the RNA polymerase itself. Double-stranded DNA enters the polymerase. Double-stranded DNA exits the polymerase. The single-stranded DNA is trapped in the center of the polymerase. One strand is the template. That's fine. The other strand, sure, that's somewhat single-stranded, but did Craig talk to you about SPIT4, 5, DCIF, stuff like that? These are elongation factors. The RNA polymerase has a central cleft, and what these processivity factors do in essence is they seal the top of the cleft. So it's a little bit like making a PCNA ring. You know, you're now topologically ensnaring the open bubble, and in fact they contact the displaced strand. So within the transcription assembly, single-stranded DNA is entirely protected within the RNA polymerase itself. And that's why I hated this model. It just seemed like complete swear words. I mean, it really was nonsense. Where you don't get these massive, great big regions. It's just, I'm trying to be politically correct and not swear. It's nonsense. It's nonsense. Nonsense. But Raj is right, because there is a consequence of transcription that's not causing a bubble around the RNA polymerase, but it is leading to underwound DNA. And that's the idea that these machines, as they move forwards on DNA, and if any of you went to John Marco's talk a couple of weeks ago, you'll have seen this already. Or did anybody go to Mike Laub's talk on Friday? Yes, okay. So, terrific talk. And he really emphasized this idea that if you've got a machine that's tracking along the double-stranded DNA, well, it's going to overwind DNA ahead of it, okay? It's bulldozing stuff apart, it's going to overwind DNA ahead of it. But you have to have compensation. And so you get underwinding of the DNA behind it. So you get negative supercoils behind the machine, positive supercoils ahead of the machine as it transits along the DNA. Negative supercoils are, by their very definition, underwound. Okay? They are more prone to melting. They are more susceptible to, for example, invasion by another molecule. For example, an RNA can come in, invade, and set up this sort of structure. Positive supercoils, in contrast, are refractory to that. So here we have our RNA polymerase trundling along. It's generating this lovely nucleic acid, which by its very definition is homologous to the stuff just behind it. It's how it was made in the first place. It was copied from it. So it's extremely prone to base pairing, and if it happens to find a meltable region, then indeed you will end up with invasion. And once you get that, now all of a sudden you get an anchor on your system, because you've tethered your transcription complex via this tail, the RNA molecule itself, and as this then rotates along the chromosome, or the chromosome rotates through it, then you've now set up a little localized domain within this region, which will actually trap the negative supercoils generated, because this is now topologically ensnared, facilitating broader melting. This is a feedback loop, okay? A beautiful feedback loop. So all of a sudden, the very act of transcription is altering the topology of the template, thank you, Raj, and creating a trap, if you like, for the RNA molecule that's held in local high proximity to the local proximity, and thus local high concentration to the region where it can invade. Potentially setting up a very unusual substrate for polymerase to engage. And this is entirely reasonable. Finally, I think this is a beautiful explanation. Andrei Kuzminov, he's in Illinois, wrote this fantastic review. I gave it to you the other day. It's the one that's got like 6 different puns in the title alone, but it's grin with the title, bear with it, and then to get into the meat of the review. I think it's an absolutely terrific overview of the ramifications of this model. But models are models, and experiments are better. Okay? Everybody can think up ideas, but how do we actually test it? And there was an absolutely stunning paper from Pura Merrick over in Washington State, testing exactly this premise using the power of bacterial genetics. So this is using Bacillus subtilis. Okay? And what Hura did was to use a very strong, rather unfortunately titled promoter, Spanky. The Spanky promoter, which is extremely robust promoter, an inducible promoter in Bacillus subtilis that will drive high levels of expression of downstream gene. And so she integrated a cassette into the chromosome and did so in 2 orientations, okay? Or 2 related cassettes, I should say. The reporter gene, and this will be either LAC-Z or luciferase, I think, a selectable marker for the integration of self. And she had one where replication was coming in from the left here, so the direction of replication, co-directional with the reporter, hence CB. And then the other one, it's a bit like Mike O'Donnell. See, if it was Mike O'Donnell, he'd have put 2 papers probably, but Hura actually put both reporters into 1 paper. Flip the orientation of the reporter, okay? So now transcription, this highly inducible and high level transcription is now counter to the direction of replication, and that's head on, HO. Okay? Then the experiment's dead simple. You just grow up the cells. You grow them up under non-inducing conditions, and you'll see why in a moment. And then you just simply switch on the expression, and you do so either in a wild-type background or in a mutant background. If you do it in the wild-type background, you can see that codirectional. Oh, and sorry, what she's doing here now is looking at RNA-DNA hybrids. You can now go to, I forget who it is, Santa Cruz or someone's catalogue, and you can look up anti-RNA-DNA hybrid antibodies, and you can buy a monoclonal antibody, S9.6, and use that in DRIP. So you've heard of CHIP, you've heard of RIP, RNA-IP, DNA-RNA-IP, DRIP. Okay, so use it in DRIP assays. This specifically brings down the RNA-DNA hybrids. Okay? And then you measure the relative amount of those, and you find for co-directional transcription in wild type, yeah, you get some. And then that's about 2-fold enhanced with the head-on. So in wild type cells, head-on setting up with this is leading to an elevated level of DNA-RNA hybrids. So they're there, but the cell's dealing with them, okay, by and large. That's using LAC-Z as your reporter, just in case there's something funky about LAC-Z. They also used luciferase or Lux. That's your quorum sensing thing, yeah, quorum sensing stuff. Not luciferase, I'm sorry. Bright stuff, shiny stuff. And again, we find head-on is problematic. But as you can see in this mutant cell line, and I'll tell you what the mutant is in a second, we see codirectional slightly elevated, but the head-on collision, the number of RNA-DNA hybrids, the extent of these hybrid molecules, these R loops, is now massively enhanced. What's the gene? Well, what might you expect to be involved in resolving RNA-DNA hybrids? An RNase H. Okay? So a ribonuclease that specifically targets RNA in the context of an RNA-DNA hybrid. Okay. RNase H3. There should be an H there. The gene is RNHC, and when you delete it, you now see massive increase, substantial increase in the level of these hybrid molecules being generated. The really, I think, mind-blowingly impressive part of this paper, and this really to me was what made it a Cell paper probably, was a series of marker frequency experiments that Hura did though. She presented this at the chromosome dynamics meeting in Italy. I was there when she first presented it. And it's so sad, scientists get so impressed with such easy things. But She showed these data, and there literally was an audible gasp from the audience. And it is just a stunning experiment. What she's doing here now is looking at the effect on genome replication, okay, using good old marker frequency analysis. The origins are at the 0 point, and the origin rather is at the 0 point, and of course at the other end, because you've artificially linearized the circular chromosome. And she's either looking at switching on expression, then looking at the replication profile a short distance later in the wild type cells. And here's our locus here. There's a slight change in the angle here, perhaps, in green. But in blue is when she switches it on in the absence of the RNase H2. We see this incredible result. And look at that vertical line. What does that mean? Why did everyone go, we still might be hard? It's a complete arrest of replication. The fork's getting here and then just stopping like a juggernaut hitting something that it can't get past. Bash. And replication just doesn't happen. So this chunk of the chromosome doesn't get replicated. The consequence, cells die. Okay? They don't replicate. It is an apparently almost permanent barrier to its replication. And again, using the Lux cluster, same astonishing arrest. When we've got the head-on orientation, when we don't have RNase H2, 3 RNase H2. Okay. So R loops are forming. Yes?
At the TER part, it seems like it's gradually replicating now. Is that what's happening?
So TER, yeah, so this is something we'll come on to in the next few slides. Again, sorry, I'm sort of putting you guys off, but yeah, so replication is coming to TER and then arresting really here, and then really it's largely flatlining. There's a little bit of noise, but I think it's mostly just noise. There might be a bit of restart events happening here. TER is the terminus of replication, and that's what we're going to talk about in the next little bit. You know, there's almost a hint of something like restarting in here. Now, what that would be, I don't know. It could be ribosomal RNA prime, that you can get R loops that actually prime replication as well. There's some evidence for those. This is, again, it's a feature that we'll come on to in a later lecture about how origins of replication might not necessarily be as utterly essential as I've been telling you for the last goodness knows how long. Anyway, head-on conflicts usually resolved by RNase H2. Yeah.
So in the 2D gels, why are the mutants so much darker than the...
Yeah, so it's suggesting that there's just a generalized arrest, so that you're normalizing based on the one end spot here, okay? So this is just telling us that the forks are just massively more enriched at this point. So it's not like there's a specific arrest point, but they're sort of just slowly forcing them with your probably grinding to a halt ultimately. We're also seeing an accumulation of some species here that can correspond to sort of regressed fork type structures as well. You'll see more of those in a few minutes. Particularly this cone here. This is something that again I'll show you in a few minutes. So it really is just telling us that within these contexts that replication is massively slowed through this site and really essentially grinding to a halt, because we see it beginning to peter out towards the bottom of the arc.
Just the wire.
Just the wires. So here they're looking purely at the region in the vicinity of the arrest site itself, so that they're not interrogating the origin of replication. And when you look at sort of zoom in on the marker frequency analysis, we can see, although here on a genome-wide level it looks like this incredible vertical line, what you're seeing is just this incredible slowing down through the region. It's crawling, eventually getting. Okay. So, R loops are, I think, unambiguously important. Back to back with that paper in Cell, Carleen Kimprick over at Stanford published another paper, a paper coming to a similar conclusion in a eukaryotic system, and Andre discusses the two of these papers in the review article. So I urge you, please do read it. It's a great review and a really amazing topic. Okay, so we've talked about transcription machinery, encountering replication machinery, and that's a problem. But of course there's another situation that can arise, and that's replication conflict, because of these two machineries converging one upon the other. And if you've got a circular chromosome and you're starting replication here, and you're bidirectionally replicating, you are going to meet at the other side. Okay? If you're a eukaryotic chromosome, you have origins here, one goes this way, one goes this way, one goes this way, one goes this way, they will converge in each other. So this situation has to happen during replication. We could call it replication conflict. We could call it simply termination. And that's when the leading strand becomes the lagging strand. The lagging strand becomes the leading strand. Okay, these converge one upon the other. And here again, topology, of course, comes into play. As we unwind, moving inwards, we unwind, moving inwards, then what's going to happen is that this region in between is going to become increasingly overwhelmed, okay? More and more positively supercoiled. What can happen with positive supercoils is that rather than being built up between two regions, essentially the machines themselves can begin to twist, okay? Wrap one around the other relative to the other. And a consequence of that twisting round here and round here is that you dissolve the positive supercoils that are getting built up here by dissipating them through the machine itself and interwinding the DNA daughter strands behind the machines, okay, in the form of what are called pre-catenates, pre-catenated intertwines. That upon completion of replication will allow this positive supercoiling barrier between the converging forks to be dissipated by passing them out behind the machines. But a consequence is now that the daughter strands themselves are highly interwoven. And obviously, cells have ways of dealing with this, and you'll see what they are. Okay, so termination in bacteria. The model system, best understood as good old E. coli, single origin of replication. And then bidirectional replication occurs. Replication proceeds around the chromosome, and eventually replication forks converge around about 180 degrees from where replication was initiated. Okay. And a whole series of genetic experiments where folks messed around with the position of the origin of replication led to the revelation that this termination site is in essence hardwired into the chromosome. We have active and dedicated termination system within the bacterial chromosome that ensures that replication occurs here and really here only. So if we move the origin of replication to here, forks will come trundling around, they'll reach this region early, but then they'll arrest. Okay? And so the genetic experiments revealed that that was the case. And it was due to cis-acting sequences in the chromosome called the TER sequences, and they're bound by a protein called TUS. T-U-S, TUS. And here we come to this important point that was just raised, that we have termination sites here, we have termination sites here. There's an array of them. We've got a whole bunch of backup sites, there's a bunch of accessory sites. The majority of termination is occurring between TER C and TER A. But what this means is that the fork that comes from RAC round this way is going to encounter TER H, pass through it, TER I, pass through it, TER E, pass through it, TER D, pass through it, TER A, pass through it, until it encounters TER C, whereupon it will stop, if another fork hasn't encountered it already. And conversely, this fork here going along this, what's termed a replicore, goes through J, G, F, B, C, and will eventually, if it reaches TER A, will get stuck there. Okay. So these are polar fork barriers. Hence the triangular drawing of them. Okay. The flat side is permissive, the pointy side is non-permissive if it's encountered. Okay. But how do you ensure this polarity? I'll tell you in a few minutes. What's the time? Oh, yes.
So, I know we have the main computer side. What's the point of having multiple ones, or is it just all in different?
These are, again, a little bit like what we saw in the eukaryotic system, where there's a sort of kinetic barrier rather than an absolute barrier. So eventually things will get through TER C, they'll make it to TER B, and it's just an additional, there's redundancy within the system. The cost to the cell of deleting these sites is very, very minimal. They really are just accessory backup sites. the consequence of deleting, for example, the TUS protein that binds to these and confers the termination in a wild type cell is actually very, very minimal as well. It's only in certain genetic backgrounds that will, again, I'm so sorry to keep doing this, but I'll come back to that later as well. There's a certain context where the TER sites become very, very important. And when you perturb that system, then loss of the termination becomes really, almost completely lethal. So that they're conditionally essential effectively. So we'll come back to the mechanism for ensuring this polarity and this directionality. But really the paradigm that's emerged within bacteria is that bacteria have dedicated termination systems. This is the reason why will become apparent again in a slide or two. But what about other organisms in that? And how do we actually map termination sites anyway? Or how do you know where a termination site is? So a little bit of a digression back into good old 2D gels, which I hope you're now brutally familiar. You've heard about how you can see the Y-arc or the bubble arcs on a gel. If you just simply do your restriction digest, you run your DNA down the gel, you then probe with a probe specific to the fragment of interest, then the majority of species within the gel will be of single unit length, non-replicating. But within the gel there will be a continuum, a smear of other species going up the gel in the first dimension, which go between 1n in size all the way up to 2n, where replication is nearly complete or the bubble is nearly complete. And the trick, of course, as you know, with 2D gels is you then tickle this stuff with ethidium bromide, you cut the gel slice out, you rotate it 90 degrees, and then you run the current down through it, and all of a sudden your linear species migrates on a diagonal. So this is now essentially a near linear species with a tiny unreplicated bit. So you see it back down at around about 2n size on the diagonal. When you've got the 3 arms of almost identical size, then this is the most maximally retarded, and so the replication fork goes up and comes back down. Okay, that you've seen before. Bubbles. Oops, sorry. If you've got replication origin, then it just becomes more and more and more circular in nature. Bubble arc gets bigger and bigger and bigger and never returns down to the bottom because it's now essentially a 2n circle. So what happens if you have termination? Well, you get signals like this, and let me take you through them. Here we have our replication fork arc proceeding, and it's reaching a robust barrier within the fork. So this species number 2 is as we replicate up, carrying on up, carrying on up, carrying on up. We reach a structure that replication, the species number 3, where replication has reached a barrier. Okay? And we see accumulation now of a signal at this site. And that's what you saw in the pausing sites. Okay? If replication then comes in from the other side of the molecule, then what we see is a rather sort of funny X-shaped molecule with an unreplicated bit in the middle. And so this is now getting bigger, it's bulkier, it's taking on a weird shape. And this is where we now depart from your classic fork arc because you're adding additional structure. So it gets bigger again, it gets more massive. And so it begins to head upwards until eventually we generate an X-shaped molecule where the replication forks have more or less converged. Okay. This now has almost 2 copies of the chromosome, 2 copies of the DNA rather. So it's about the 2n position, but it's way up there in space because it's got this really structured X shape up there. Okay. And so if you think you've got termination, you could do 2D gels looking at the regions. Oh, sorry, that's specific termination. Okay, beep, 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 beep. What about stochastic termination? If rather than terminating at a specific site, you've simply got random fork collisions over a fragment. Well, in this case, forks will progress in different distances. Forks will come in from the other side at various different points. And so at all these different positions along the fork arc, you can begin to get these additional, more massive species being generated. These gradually begin to fill out. And because they're happening across the entire continuum of the replication fork arc, essentially you get this sort of fuzzy cone-shaped sort of shape, yeah? Cone-shaped shape. This is just an amalgam of all these different species blurring one into the other, giving you a sort of random fork collision signal. So if you do exactly this experiment, this is done by one of my postdocs a few years ago now, quite a few years ago, looking at the bacterial system and probing for the TER-C region of the chromosome, Then we have our one end spot, we have our Y-arc, we have our robust replication fork block at ter C, and then emergence of this sort of little drumstick, which is the structured molecules. And again, just taking you through it. We have arrest of the fork at ter C. We have convergence of the fork adding extra structure, giving us the beginning of the drumstick, getting further in and further in. Okay. So if you're really masochistic, what you could do is a whole series of 2D gels looking to see, can we detect signals like this that correspond to replication termination within a region of interest? Okay. So in E. coli we have this robust barrier to replication, this robust directional barrier to replication. How does it actually function, and how is it directional? It's an extraordinarily elegant way in which this is done. And this was a series of biochemistry and also crystal structures from Nick Dixon's lab in Australia, where they took the classic TUS binding site, which is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 nucleotide drag for TUS, plus a variable nucleotide at the end. But what they noticed was that in addition to the core footprint of the TUS protein, invariably adjacent to where the protein itself bound, you had AG on one strand, which of course is a CT on the other strand. Mutational studies showed that the C was of pivotal importance for the function of this trap. And what the crystal structure showed, and I'll show you the structure itself in a second, was that what happens is as DNA B is moving 5' to 3' along the lagging strand template, it's unwinding DNA. That's what it does. It's a helicase. It unwinds DNA. It does so by steric occlusion. Okay? So it's knocking off essentially the leading strand template ahead of it, generating single-stranded DNA. It melts out this GC base pair, giving you a single C residue adjacent to the binding site for TUS. And as you can probably guess from the structure that I've drawn here at rather low resolution, is that TUS contains a binding site for this C. Okay? It locks into this binding site, and in doing so massively stabilizes, by about 2 orders of magnitude, stabilizes the binding affinity of TUS for this melted DNA, and essentially locks down the structure. So if DNA-B approaches from this side, the very act of melting itself stabilizes the TUS-TER complex and makes DNA-B unable to pass through this barrier. If it approaches from the other side, well, it doesn't have the binding pocket. It's an asymmetric molecule, and so it simply knocks TUS off. It's that simple. Extraordinarily elegant. And here indeed is the co-crystal structure of TUS bound to a TER site with played duplex at the beginning of it, and there's your C sticking into the little binding pocket here. And through a series of mutational studies, both in vivo and in vitro, Nick has proved this model beyond any shadow of a doubt. So that's how you do at least in the bacterial system, you do the polar arrest. Because this will stick here, but coming in from the other side, it will melt it away quite happily and then just meet in the middle. Okay. So the dogma until, rather annoyingly, until very recently, where it now looks like Pseudomonas might not have a termination system, is that bacteria have dedicated termination systems. Dedicated termination is a universal phenomenon in bacteria, was what we said until relatively recently. Bacillus subtilis has termination sites. They've been mapped genetically, they've been mapped physically. They exist, they happen. They must be conserved. Clearly, it's a conserved mechanism. Turns out Bacillus subtilis uses an entirely different way of an entirely different protein to mediate arrest. Slightly more complicated. It's a bipartite site. It's got an A site and a B site, or subsites, if you like, within the termination site itself. But again, these are directional. Okay? These are polar, even though both of them bind the dimer of the RTP protein. So the protein binding, at least at the macro level, looks kind of symmetrical. Dimer of RTP binds to A, dimer of RTP binds to B, and there's a little bit of cooperativity between the two dimers binding to form essentially a tetramer locked down in the DNA. And the really cute thing is that where the asymmetry comes in is that the A site is a low affinity site, the B site is a high affinity site. So if the fork comes in here, then the binding of RTP is comparatively weak. It's relatively easy to displace this low affinity bound site, which will fall off, and then it can truck through the high affinity site. However, the high affinity site, if approached from this side, because of the cooperativity with the lower affinity site, just gets that little bit extra bit of stability. Okay? So the fork can't push in from this side, but can push in through this side. So totally different mechanism, totally different protein, evolutionally very distinct organisms, but the net effect is the same. Permissive site, non-permissive site, directionality. Okay. Why do bugs like to have active termination systems? I think the answer lies in their lifestyle. The microbiologist amongst you will know that bacteria don't do mitosis. Bacterial chromosome segregation occurs during the very process of replication itself. Okay. Eukaryotes replicate, they have a G2 period. During that G2 period, sister chromatids are cohesed, and eventually in mitosis, they resolve. Okay. Bacteria are smarter than that. They don't waste so much time with the silly G2 phase of the cell cycle. They get straight down to the business of replicating and dividing. They got to live, and they got to live fast. And so what they do is they actually begin to partition the chromosome into what will eventually become the daughter cells during the replication process itself. So the origins are replicated, they're segregated. There's a little bit of a delay. The delay is due to the resolution of hemicapnium. But there's a slight delay. But eventually, as they're being replicated, you're actually bundling the chromosomes up. Okay? Those of you that have been to any of the talks by Jindan Wang in the micro department, she's studying this process in essence and doing some really amazingly beautiful work with it. The mechanisms are being resolved as to how it happens, largely in her lab actually. But the phenomenon is clear that we have segregation concomitant with duplication. And that's great, that's a really, really quick and efficient way of doing things. But there's a problem, because most bacteria have chromosomes that are circles. Okay? And if you have an odd number of homologous recombination events between two circles, then that's going to generate a dimer. Okay? So you get a dimeric circle as cartooned here. And this happens in about 6% of cells per generation. Well, it's been measured. If you try and segregate through this, then you are in deep doo-doo, I think is the technical phrase. Because you simply can't get a septum through this DNA, or if you do, you're going to rip your chromosome into two halves. Bacteria, of course, are smart. They have undergone many generations of evolutionary pressure, and they have a system which resolves dimers down to monomers. There's this little red triangle here. There's a thing called the diff site. It's a site-specific recombination system. A specific recombinase, a thing called XERCD, binds to the diff site and catalyzes site-specific recombination between these two sites to generate two circles. Okay. So you generate circles, monomeric circles, from a dimeric circle. XER, C, and D are the molecules that do it. And that's beautiful, and it's fantastic that it works. But of course, the only way it can possibly work is if the XERCD system are acting at the very last replicated region of the chromosome. Okay. Because you need to juxtapose the sites in order to carry out the recombination reaction. It'd be absolutely flipping useless to have recombination sites up near the origins of replication, because they're now well, a micron and a half apart. They're physically about as separate as you can get within the nascent mother cell. So you have to have diff site in the last replicated region of the chromosome. That's why you have termination systems, to ensure that this region between ter A and ter C is the last replicated region of the chromosome, because that's where your diff site is. Okay. You segregate everything else. You have that one last little bit. If you have a dimer, you resolve and you segregate. Yeah. Just going back a few slides, you'll see the diff site in E. coli is between ter C and ter A. Here it is in Bacillus subtilis, again, between in the vicinity of ter 4 and ter 2. So again, it's held at the last replicated region of the chromosome. And folks have done experiments where they've moved the diff site to different locations, and it doesn't work anymore. Okay, so the position is absolutely pivotal to its function. So that, I think, is why you have dedicated termination systems, is to ensure this. And sure enough, again, the genetic experiments say that a TUS mutant on its own, that's all right. A diff mutant or an XER mutant on its own, they're okay. Put the two together, synthetic lethality, or at least synthetic extremely sickness. So what about archaea? Bacteria are nice and simple. They've got one origin of replication. Forks come out, they converge, probably most of the time just naturally around about 180 degrees around the chromosome. But every so often, they'll slow up, and that's why things need the ter system to allow things to be held in the last position, forks to be held in the last position. But good old friend, Sulfolobus, has 3 origins of replication, yet a simple circular chromosome. So does the same logic apply? Circles, dimers, resolution, terminus, blah, blah, blah. We didn't know. And so Ian Duggan, the postdoc in my lab, extraordinarily masochistic Australian guy, decided he would do, I think it ended up being about 150 2D gels. He spent about a year and a half in the cold room running 2D gels. I didn't make him do it, he volunteered to do it. And just surveyed across the regions between the origins of replication, looking for termination structures. It was the ultimate disappointment. He looked and he looked and he looked for dedicated fork termination sites, and he couldn't find any. All we could see were evidence for stochastic fork collision. So we thought, well, maybe that's just the case between these 2 origins of replication. Back into the cold room, bunch more gels. Nope, same here again. In this site as well. Random fork collision, not dedicated termination system. And finally, of course, for completeness sake, we had to do the 3rd set. And again, a bunch more 2D gels, frostbite setting in by this point, poor guy's vaguely bluish grey in colour and just not terribly happy. Stochastic fork collision. Okay. No dedicated termination system in Sulfolobus. But this is brutal. I mean, this is horrible. He shouldn't have had to do this. I mean, I'm a swine for making him do this sort of thing. I really am. Horrible person. But he did volunteer. Because there's an awful lot more elegant way of doing the experiment. And that's to just reprogram the chromosome itself. The trouble was, Ian came into the lab before we could do genetics with Sulfolobus, so we were just about able to do genetics with it. And then Rachel, who lectured to you a wee while back, we collaborated with a lab in Copenhagen, and we learned how to do genetics properly, and we made a series of mutants that she probably talked to you about, where we took out individual origins of replication. If we look at the marker frequency profile of wild type cells, we have RAC2 at the 0 position, RAC3, RAC1, so the 3 peaks. The troughs correspond to where replication forks converge, the termination sites, if you like. So what happens if you take out an origin? Or take out 2 origins? Why not take out 2? Does termination stay at the same place? In which case we would have dedicated termination systems that are hardwired into the chromosome, or do they reposition? So here's the experiment. We took out RAC2 and RAC1, leaving only RAC3 actively replicating the chromosome, single peak here. And very pleasingly, the sites that were formerly used as termination sites are no longer used for termination. Rather, the trough occurs at a new position previously not a termination site and is 180 degrees around the chromosome from the single active origin of replication. So in 2 experiments in the course of about 3 weeks, Rachel was able to confirm what Duggan had taken about 18 months of shivering in the cold room. There is not a dedicated termination system in Sulfurobis. Rather, we have stochastic fork collision. Forks simply collide halfway between active origins of replication. Why? Why is it different from bacteria? I'll tell you in a later lecture. What time is it? Yes.
So if you like, I guess like mess with the rate of replication on like one side of the origin,
Yep.
Cephalobus, would that also move the termination?
Probably would if we could stick a barrier in, some
Yeah.
sort of fork barrier.
Mhm.
I used to be, well, I'm still horribly smug in a lot of ways, but I used to be particularly smug because obviously we work in Sulfurobis, it's fantastic, it goes 80 degrees centigrade at pH 2, you take Elber off, you add sulfuric acid, you heat it to 80 degrees, contamination, we don't care about contamination, smug, smug, smug. Then actually what Ian wanted to do was to do exactly that experiment. He wanted to take a termination system, slap it into the chromosome, find out what happens, can we make a conditional rest site, what happens with the rested forks and all that sort of stuff. So where do we get a termination system from if there isn't an active one in the organism itself? Well, there are plenty of hyperthermophilic bacteria.
Mhm.
And so he said, oh, well, let's get the Tertus system out of, I think it was Bacillus stearothermophilus or Geobacillus stearothermophilus. So yeah, great, good, do it, go ahead. We'll stick into the chromosome, see what happens. What I hadn't realised was the silly bugger went ahead and he actually ordered the organism, rather than just getting some genomic DNA or making the genes synthetically, he bought this bloody organism. Fine, which is great. The flaming thing sporulates. So he grew it, he kind of abused it, he stuck it in the fridge, and suddenly there's these Geobacillus spores everywhere. And it grows at 75 degrees and it likes acid, and all of a sudden, for the first time in existence, we had contamination in our Sulfurobis plates. It was horrible. We thought for a moment, oh my god, we've got morphological mutants, it's rod-shaped, this is fantastic. So anyway, it turns out Geobacillus doesn't grow at 78, but yeah, I mean, Duggan was fantastic, it was a wonderful postdoc. I'd really kick his arse for that, I tell you. So yeah, if somebody wants to come into the lab, you do that experiment. He never actually got around to completing it because he ended up getting this termination stuff working really nicely, and stuff I'll talk about later, but we found that there is actually a diff system in Sulfurobis and all this kind of stuff. So that project is hanging there. So if anybody wants to come to the lab. Sorry, I get kind of worked up a bit. I'll just very briefly start this bit, or at least introduce this bit to you, okay, and then we'll wrap it up for the day. So in eukaryotes, the available evidence says that it's also stochastic fork collision that mediates replication termination. And to some extent, this makes a heck of a lot of sense. If you've got an organism, whether you're an archaeon or a let's draw a chunk of chromosome and let it either be circular or linear, depending if you're a archaeon or a eukaryote. You've got an origin here, you've got an origin here, you've got an origin here, and you've got a termination site here, and you've got a termination site here. Well, that's all well and good if your origins all fire actively. Okay? Because if you have inefficient origin firing, let's say this one fires, maybe the fork can get through this barrier, it'll encounter this barrier and stick. Similarly, a fork coming through here. Dedicated termination systems have the potential, if not all origins fire, to lead to unreplicated regions of the chromosome. And as a consequence, it really would be bad evolutionary sense to have dedicated termination systems between every origin and replication. And so I think that's partly what we're seeing with Sulfurobis. There's another rationale to it. I'll talk about it in the final lecture. But just a priori, it makes sense that there should be the plasticity in the system to allow for not every origin to fire at every cell cycle. Otherwise the cells wouldn't replicate the chromosome and they'd cark out. But how does it actually work? What happens to the forks themselves? And this was a It's simply another stunning paper. There's a guy called Johannes Walter at Harvard Med. He does beautiful science. His data are always just as clean as they could possibly be. And of all things, he studies Xenopus. I mean, stinking great big frog. He does stuff with crude extracts as well. I mean, he just does brilliant experiments with crude extracts from Xenopus that support replication. And the genius is not so much the refining of the system, but just the purity of his experimental design. I quite like his work. And it's such a neat idea. Xenopus is a pain in the butt in some ways because it's egg extracts you're dealing with. They'll replicate anything. You give them a bacterial plasmid, they'll replicate it. But they'll do so in a controlled, once per cell cycle way. So it's a coordinated, controlled system, but it's origin independent. Anything will work to assemble replication forks. So how on earth do you study the process of replication termination if initiation can be randomly occurring anywhere within the molecules that you're studying? And brilliant, simply brilliant idea. He took an array of lac operators, so binding sites for the bacterial lac repressor, and he stuck them, there's about 16 copies of the lac operator on a otherwise 3 kilobase plasmid. And you bind LAC-R, so you've got this chunk of the plasmid now that's coated with the lac repressor. Okay. You then take your Xenopus egg extract, you stick in a bit of radial label, then DATP, and you let replication start. It'll start here, it'll start here, it'll start here. It doesn't matter where it starts, because ultimately what's going to happen is one fork is going to converge on the right-hand side of the lac array, the other one will converge on the left-hand side of the lac array. Okay. And so you end up with these stalled replication forks. How do you get rid of lac repressor? You chuck in a little bit of IPTG, you induce it, it falls off. And then you can follow this population of molecules. It's more or less instantaneous. The minute you add IPG, it falls off. It's a very rapid allosteric effect. And so you can then watch the synchronous movement of the forks into this unreplicated region as they converge one upon the other, and then ultimately lead to termination. So what he does here is simply add IPTG or not at various times. So in the absence of IPTG, where are we at? Let me just so in the absence of IPTG, then you have this species here. So what he's doing is he's taking the partly replicated molecule. It's got a single XMM on site here, okay? So it linearizes here, it linearizes here, and if you just open this up, it gives you an X-shaped molecule, okay? You add IPTG, you essentially generate these double Ys as they converge one in the other. So these are structured molecules. Ultimately, you generate linear molecules where the leading strand and the lagging strands converge. And finally, what you're hoping to see are covalently intact daughter molecules shown in the red. So you'll convergence of leading and lagging strand, and eventually ligation event. Okay? You could follow the presence of these in 2D gels, but you don't need to, because what you can see is that the double Y-shaped molecules have a slow migration. The linear molecules have a faster migration under a standard gel. So you can take IPTG, in the absence of IPTG, then you've got this robust arrest of these double Y arcs, very few linear species. If you add IPTG and follow it for 5.5 minutes and so on, then you see the disappearance of the double Y molecules and the reappearance of the linear molecules, okay, as replication completes and reaches its culmination. That's looking at double-stranded DNA. You can do exactly the same experiment, but just simply denature the strands and run them in an alkaline agarose gel and look at the single-stranded DNA. And what we see is really just looking at the 2 kilobase fragment here. Okay, that's the 2 kilobase from this arm. It gets a bit bigger, and eventually it will converge with the leading strand, sorry, the lagging strand on the other side to generate a covalently intact 3.1 kb molecule. And we see exactly that happening here. Starting out at 2 kilobases, getting bigger, 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 giving us our product molecule. So we can follow the kinetics of the completion of replication and ligation. Yeah?
What is the last, so it goes like 8, 10, 8? What is that, why is that
That's
last
8 minutes in the absence of IPTG, and so it remains
Oh.
with the arrest site there. So it's just confirming that over the time course of the experiment, it's not just that you're kinetically bashing through the lactic compressor. So at 5 minutes is when they add IPTG, so that's your start point. You see this gradual disappearance. By 8 minutes, then essentially it's all gone in the presence of IPTG and everything's resolved the product. In the absence of IPTG, well, you are generating a little bit of product. Eventually this is beginning to bash through. And that's probably the 2 years of postdoc work that you don't hear about, was them getting exactly the right concentrations or relative of extract and plasmid, following the kinetics just precisely enough to be able to parse out the various differences. You know, it's beautiful when it's that one clean figure of nature, but I suspect it speaks to a lot of postdoc pain in this generation. And then the final complication, and I'll just do one more slide after this, then we can stop, is that one can also look at the topology of the daughter molecules. So you'll start out with this, you'll generate convergence to give you this sort of theta-shaped molecule, and then eventually, if these do actually complete replication, then you'll have the 2 daughter molecules held to one another. This is the pre-catenanes, now becoming full catenanes because replication has been completed. They're pre-catenanes because replication is ongoing. Once replication completes, they become catenanes. So they're just interlinked circles, okay? And then ultimately, what we expect in a system is that the cats should resolve down to monomeric circles. I just noticed they've got a typo in Nature, of all places. It's a circular nonomer. It's actually a monomer. That's shocking. They should retract it based on that alone. And they see, basically now looking at the topology, they see generation of the theta molecules, the cats, and then eventually resolving back down to supercoiled circles. Okay, so this is just simply resolving the various different topological species again in an agarose gel system. They do this enough times, they can actually quantitate the amounts of the various products, and what they see is dissolution occurs first. So in other words, the 2 molecules converge one upon the other, and then to all intents and purposes replication is complete. But ligation itself, so covalent integrity, is imparted a little bit later. So first of all you have dissolution, then you have some nicks left behind that are sealed up, and then finally, kinetically, the final event is catenation. So we have this linear progression of events taking place, recapitulated beautifully in this in vitro system. I'm trying to remember how many slides I have left. Yeah, no, there's enough that I wouldn't go on there. So with this system in place, they can now address the questions of what actually happens to the forks. We have the kinetics of what's occurring, but what are the molecular events that are occurring concomitant with these changes in the DNA? Which proteins are on the DNA, which are not, which have left, which are brought in and active. And that's what I'll pick up on for the first 10 minutes or so on Wednesday.
Is that last paper one of the papers that's posted on Canvas? The one that you were just going over?
I haven't put the actual primary data on.
Okay.
There's a review article by Walter and Walter, though, that really discusses a lot of this data.
Okay.
And also there's stuff will come on to on Wednesday as they then look at the kinetics of the disassembly of